cover some of the bovine diseases. Now looking at this, what does it tell you?
There's blood.
Well, yes, there is blood.
That's all I
But
got.
what's about this blood?
Yeah, it's pretty red.
Is it bright? So if it is bright, it's coming from where?
Lower intestines.
Okay. And is this profuse or is this,
It's not focal.
so is this serious or not?
It's pretty serious.
Blood is always serious.
Okay. And is a larvae. So that's the property of coronaviruses. Now when you hear coronaviruses, there are two things associated with them: respiratory and or enteritis. So respiratory or digestive diseases. Now there is another class of viruses which are toroviruses. They have very similar also symptoms, but we're not interested in them. So coronaviruses, they're RNA viruses, and they target enterocyte, crypts, and cell of the respiratory tract. So then, if they target those locations, the expectation is that we will have a problem with diarrhea, necrosis, and we'll have a problem also in the respiratory tract for the respiratory symptoms. The virus bovine coronavirus 1 is associated with the three syndromes: cough diarrhea, winter dysentery, and respiratory disease. Now, the cough diarrhea and respiratory disease, they can be both present in the animal. Winter dysentery can be associated with respiratory symptoms. Now, winter dysentery is the most serious one because of the explosive diarrhea, which is associated with the blood. The morbidity is about 20 to 100%, mortality, though it is low. In the respiratory disease syndrome, there is the usual respiratory symptoms: rhinitis, discharge, conjunctivitis, and so on. And when it becomes more serious, if it is associated with bacterial infection, causing what's called the shipping fever pneumonia. And this is the bacterium is Manamia. It's called Manamia haemorrhagica. Okay, so the pathogenesis, the virus multiply in the mature cells of the intestine, also in the crypts, causing the death of the cells. And as a result, you have the imbalance in electrolyte, the hemorrhage, which is obvious. And then following this, obviously, what you expect is hypoglycemia, is acidosis, hypovolemia, and so on. Those, symptoms or those features are associated usually with diarrhea. So we said if the respiratory symptoms are complicated with bacteria, Manamia haemorrhagica, then what you will have is the severe disease. Now the reason it's called shipping fever is because of the stress as a result of shipping animals. And that stress predispose the animal to bacterial infection. For diagnosis, the virus can produce cytopathic effect in cells. So cell culture can be one of the methods to isolate, if you want, the virus. And because the virus is present in secretions, whether it's respiratory tract or in feces, then it makes sense to opt for those tests that detect the virus or viral antigens. So we have ELISA for specimens such as feces and nasal swab. You can use RT-PCR. In this case, they use pan-coronavirus-specific primers. Remember, this is RNA virus, so it mutates, and there are a number of variants. So they can design pan-coronavirus-specific primers that can identify these variants. Immunohistochemistry also is used, like immunoperoxidase that we explained in the lab, can be used also to identify the virus in tissue. Now, in relation to immunity, there are vaccines, and the vaccination is usually practiced during calf suckling period. Why do you think it should be practiced during the suckling period rather than before? Okay. Say that again?
Like stress from weaning them could predispose them to getting it?
Okay, but that's not the actual reason. The actual reason that the animals have the virus already, have been exposed to the virus. So the virus, if you want, is a normal inhabitant in those animals. However, it only causes diseases when there are factors such as the stress, immune suppression, and so on. So because they are exposed to the animal, it means they have a memory response. And a memory response means when you boost the animal, you'll get immediately, within a few days, even a couple of days, you get antibody passed through the colostrum to the immunity. Yes?
So, if you were to do a vaccine regimen, wouldn't you still want the dam to be vaccinated at a certain point when she's preparing to give birth, rather than just depending on the booster after?
That's another option. Another option is before coitus, you vaccinate the animal. So then you make sure that, it has, and particularly with live vaccines, because these will last for a long time, and immunity will pass, to the calf. Another way also, the third way, is to vaccinate pregnant animals. Now you vaccinate pregnant animals just before delivery, few weeks before delivery. And obviously that has to be not a live vaccine that can cause abortion. So most of the cases they vaccinate with a killed vaccine. Okay, generally speaking, for respiratory disease and winter dysentery, there are no vaccines. However, one can use attenuated bovine coronavirus vaccine to reduce winter dysentery, the effects as a result of winter dysentery. This is a serious disease which is reportable. It causes immune suppression, congenital defects, abortion, diarrhea, and hemorrhages. This is a very serious disease. It's present in the US, and as I said, is reportable, and so many questions inevitably come in relation to this disease. So as shown here, you could see the necrosis, ulcer and necrosis in the mouth. You could see the severe necrosis in the intestinal epithelium, and this is the esophagus bleeding, ecchymotic hemorrhages in the esophagus. So, what disease is this? Bovine viral diarrhea. Now, bovine viral diarrhea belong to the family of Flaviviridae, most of the members of the Flaviviridae are carried by insects, except bovine viral diarrhea. It's not carried by insects. In fact, the name can be misleading in relation to the route of infection, because the route of infection in bovine viral diarrhea is respiratory rather than oral. This is again an RNA virus, and it causes immunosuppression because it infects monocytes, polymorphal nuclear cells, PMNs. It replicates in lymphocytes as well, and causes thrombocytopenia, hence the presence of hemorrhage. There are a number of scenarios, or if you want, symptoms associated with this disease that ought to be really known very well. The genus pestivirus, and that virus has two genotypes, and each of the genotype has two biotypes. Okay, so when we say genotypes, that means in terms of the sequences are, different, but they're coming from the same virus. And when we say biotype, it means that biologically, each of the biotype can cause a different outcome. What is BVD? It's an acute, typically episodic worldwide disease of cattle of all ages, but usually occurs between 6 and 24 months of age. One of the most important outcome of this disease is the presence of persistently infected calves. And as a result of the persistent infection, as a result of this, the spread of disease is very fast. Hence, controlled management, removal of persistently infected calves is very important. The virus can infect the fetus, and infecting the fetus can cause teratogenesis. And basically what this means is the congenital defects. This is also serious enough, and we're going to see when it causes congenital defects. So the virus into the respiratory route, replicate in the nasal and nasal mucosa and tonsils, arrive to the regional lymph node. From there it becomes systemic. It reaches the intestine and infect the intestinal cells. Most of the cases are subclinical. However, when it causes a disease, it causes symptoms, it causes ulceration in those locations, including the intestine. Hence the hemorrhages are present. And if it causes thrombocytopenia, then also the hemorrhages are accentuated, edema and hemorrhages. This comes inevitably quite often. So what are the clinical features of this disease? The first one is postnatal infection in non-pregnant young cattle. So non-pregnant young cattle. There is the mild form and severe form. In the mild form, what you basically see is the leukopenia with the nasal discharge and diarrhea. In a more severe form, now you get the thrombocytopenia and the hemorrhages as a result of this, edema and hemorrhages. The second, if you want, clinical presentation is infection in pregnant cows. And these numbers are important because they come also in the exams and in NAPI. If the infection occurs very early during pregnancy, usually it results in abortion or perhaps resorption of the fetus. If the infection occurs between 100 and 125 days, and sometimes it's reported 40 days to 125 days, it results in the persistently infected calves. So we need to know when we have persistently infected calves and when we have congenital defects and when we have the other signs. After 125 days, then what happens that the fetus now or the embryo will build its immunity, its own immunity, and then it can control the virus. So therefore, as a result of this, the newborn will be okay. Some of them have some defect, however, they're not serious enough to impair with their survival. Now, as we know, in cattle as well as in swine, the transfer of immunity of immunoglobulin does not occur from the mother to the fetus. And because of this, the newborn is completely dependent on immunity from the mother. However, the embryo can produce its own, if you want, immunity, but that immunity is very preliminary, or if you want, very poor, because it's only the viruses that it encounters during pregnancy. So the viruses that are exposed outside, that are outside, the newborn cannot resist their infection. As a result of this, they are completely dependent on their mother immunity. Okay, so although abortion can occur at all stages, the most common is the first 50 days of gestation. Now we're going to talk about mucosal disease, because that's the other aspect of, that's the, if you want, the advanced stage of bovine viral diarrhea infection. The mucosal disease is very serious because obviously the lesions that it causes, whether they are ulcers, necrosis, and hemorrhages. We will understand how, we can understand this better in this picture. If the pregnant dam embryo is infected with the non-cytopathic bovine viral diarrhea virus. So bovine, these are the two biotypes. We have the cytopathic and non-cytopathic viruses. So what this means, one causes cytopathic effects when it's grown in cell culture, the other one doesn't cause a cytopathic effect. Not only in cell culture, but also in the embryo. So the non-cytopathic virus, if it infects a pregnant animal's embryo, then what it will result in a persistently infected calf. But if this persistently infected calf is then is infected with the cytopathic virus, then this will cause a mucosal disease. So mucosal disease can occur only when the two biotypes infect, the embryo earlier and later on the newborn calf. If the embryo is infected instead of non-cytopathic with the cytopathic virus, then you have the congenital abnormalities. This can involve the eye, the brain, etc. And if a normal calf is infected after it is born, with the cytopathic DVD virus, then where you get virus diarrhea or persistent infection. So those are, if you want, the presentation, the clinical presentations that occurs following infection by either of the biotypes or both biotypes.
Dr. Sharp,
Now, yes,
could you please explain that again?
okay? So, in the top here, what you have is the embryo, which is infected with non-cytopathic virus. Okay, so that's the one of the biotype of the virus. After it is born, if it is, what will happen is that this calf will shed the virus persistently. So that means that become a problem for other, calves in the vicinity or in the same location. But if it happens that this persistently infected calf is infected with the cytopathic biotype, then this will result in the mucosal disease, the ulceration that we find, necrosis, and so on. Yeah.
How would it then get infected with the cytopathic BVD when it's already born?
Well, it can get infected by a virus
Okay.
outside.
Okay.
Yeah. And if the embryo was infected with the cytopathic version or the biotype, then what you will have during pregnancy, the abnormality get developed because the cytopathic is more severe to the non-cytopathic in relation to infectivity. So it causes these congenital abnormalities. But if the calf is born and there is no, it's not shedding the virus and it's not